This is a production funded by viewers like you. Transcription in Prokaryotes. Prokaryotic transcription occurs in 3 different phases: initiation, elongation, and termination. In initiation, transcription factors bind to the promoter sequence on the DNA. The promoter sequence is between 35 and 10 base pairs up from the initiation site. When the transcription factor binds to the DNA, it allows RNA polymerase to bind to the promoter site. Once RNA polymerase attaches, the DNA unwinds and is now ready for elongation. In elongation, the DNA that has been unwound by the RNA polymerase can then be replicated. RNA polymerase moves from the 3' end to the 5' end of the template strand, or downstream, and attaches free nucleotides to the strand. This forms a new strand of mRNA. In termination, RNA polymerase reaches a terminator sequence and is bumped off the template strand, thus ending the 3 phases of transcription, and leaving a new mRNA strand to move on to the next step, translation. Now we'll move on to operons. Genes are not just randomly ordered on the double helix. They're actually organized into operons. Operons are clusters of co-regulated structural genes. They consist of a promoter site, an operator site, the structural genes, and a terminator sequence. For transcription to begin, RNA polymerase must bind to the promoter. Now this will only happen if the correct or corresponding transcription factors are already bound to the promoter site. Here you can see these are the transcription factors, and this is the RNA polymerase. RNA polymerase can get knocked off the DNA if a repressor protein is already bound to the promoter site. Repressor proteins inhibit RNA polymerase movement across the operon. For a repressor protein to be present, a regulatory gene that codes for said protein must be turned on. These genes are not necessarily located in close proximity to the operon, but can be anywhere on the genome. As you can see here, here's the operon, and here's the regulatory gene, or the gene that codes for the repressor protein. If the regulatory gene is turned off, then a repressor protein is not transcribed and therefore is not present. If there is no repressor protein present on the operator site, RNA polymerase continues transcription of the structural genes. Here's RNA polymerase. It moves down, transcribes the structural genes. And it does so until it reaches the terminator sequence. Once it reaches the terminator, it detaches from the DNA. Now there are 2 different types of operons: inducible operons and repressible operons. Let's start with inducible operons. Inducible operons are typically in the off position, meaning that a repressor protein is usually bound to the operator site. The operon is only induced or turned on when the repressor protein interacts with a specific molecule. Here you can see the interaction. There is a shape change. The repressor protein detached from the operator site, and the operon is turned on. Here's an example of a inducible operon, the lac operon. Now you can see the lac repressor. It's attached to the operator site. When lactose is present, the lactose binds to the repressor protein, and the repressor protein releases from the operator's operon. And then RNA polymerase is free to complete transcription. The other type of operon is the repressible operon. Repressible operons are typically in the on position. This means the structural genes in the operon can always be expressed. They can always be expressed unless a repressor protein attaches to the operator site and turns the operon off. This means that RNA polymerase cannot bind to the operon. An example of this is the action of an operon when tryptophan is present. If tryptophan is present, tryptophan binds to the repressor molecule, and the repressor molecule can then bind to the operator site and prevent RNA polymerase from binding to the operon and transcribing the structural genes. As you can see here, the repressor molecule and tryptophan work together. They are co-repressors, as was the last example with lactose. Examples I just talked about are classified as negative gene regulation. Negative gene regulation entails the switching off of an operon by an active repressor. Naturally, there is a way to positively regulate gene expression through positive gene regulation. In positive gene regulation, activators bind to operator sites and turn on the operon, thus allowing transcription to occur. Thank you for your attention.